I'm just going to remind everyone that relatively low telomerase and short telomeres in humans appears to be a tumor suppressive mechanism early in life, but causal for disease late in life. And this is something that's commonly used in the evolutionary field to be considered as antagonistic pleiotropy. And this will come back up in the area of what the cancer risk might be with respect to telomerase activation. So this is just the schematic of what you've seen already. If I can move this forward. This area of the schematic is the telomerase, and you can see the template region of the RNA component is what dictates the synthesis of the telomere sequence TTA GGG. And as Bill and Mike and others had mentioned earlier, this is basically the primary mechanism by which telomeres can be elongated. And then this area is the telomere, and it's much more complex than just that TTA GGG. There's a large number of proteins that bind the telomere, and the more of those proteins there are, the longer the telomere, the greater the mass of telomere binding proteins, and that it appears to have an inhibitory effect of telomerase being able to bind to the very end of the telomere. And this is what contributes to what is sometimes called the equilibrium set point for telomere length, which we all have basically set by our personal genetics and potentially lifestyle. And then this is just another picture, a little more detail of what might be happening at the single-strand overhang in terms of proteins that are known to inhibit the ability of telomerase to bind to the end of the telomere where this extension is going to happen. And that helps keep telomeres from getting too long. Many people have asked about what regulates telomere length, and it's a combination of many of the proteins that bind the double-stranded region as well as this overhang. And there's a schematic here. Just normally you see these ends just highlighted as a colored blob, but there's a lot of detail and a lot of work done by many other scientists in this area to understand the exact mechanisms of telomere maintenance and the role of these proteins in protecting chromosome ends. And when the telomere gets critically short, some of these proteins can't bind, and that uncaps the telomere and allows all of those negative things happen that have been mentioned earlier in terms of triggering DNA damage response and ultimately cellular senescence and loss of tissue function. So what we know now is that either unfortunate genetics, if you have mutations that reduce your telomerase activity, or a lifestyle that's not conducive to telomere maintenance, you have a vicious cycle essentially where low telomerase and telomere shortening can of course lead to all of the things that you've heard about: accelerated aging, cell senescence, inflammatory damage, ultimately loss of cell and tissue function, and degenerative disease, including cancer. But these conditions themselves can accelerate telomere loss. So it is very important for us to make an effort to find molecules that can break that vicious cycle, increase telomerase activity, and help maintain healthy telomere length. And then I want to mention an interesting threshold effect which shows up in some of the science studies that I'll describe in a little bit, where there is not much of a phenotype for telomere shortening until you get down to a critical length of telomeres. And Bill put this number around 5 kb. I put it lower because the conventional markers for measuring telomere length have maybe 2 or 3 or 4 kb non-telomeric DNA that's not accounted for. So the length where you might start seeing cellular senescence, we believe, is closer to this length, and you need a different type of assay to see that. But once telomeres get to that threshold length, and it may differ between individuals, you see massive genomic instability. As the chromosomes become uncapped, they can recombine with other short telomeres, causing a cycle of what's called fusion bridge breakage and genomic instability. So there's, in this period here, this timeline going forward, which can be just chronological age or number of cell divisions, it's influenced by a variety of stressors, there's increasing risk of disease. So it's actually useful to measure telomere length in that period to be able to identify subjects who may need to have more frequent monitoring, better treatment, potentially telomerase activators like TA65. And once you get below this with an increasing fraction of senescent cells, then you have very often frank degenerative disease, including cancer. Now, most people think that telomere length just shrinks monotonically in a uniform manner. When you do big population cross-sectional studies, of course, it looks like that, but on an individual level, it's now emerging that telomere length is quite dynamic, and you can pick up significant changes, statistically significant changes in telomere length, either rapid growing or rapid loss within just a few months period. So this kind of oscillation and maybe this equilibrium that goes up and down is important biologically or clinically. We don't know yet, but what it does mean is that you shouldn't just rely on a single telomere length measurement to assess your disease risk or your health status. Very likely we need to have multiple measurements and find the average telomere length in a short time period. And we do multiple measurements over short time periods to get a better estimate of the true telomere length in a subject. But if you have multiple time points over 3 months or 6 months or annual visits, you'll have a much better estimate of the true rate of loss of telomere length within your body. And what's emerging is that the rate of loss of telomere length or the increase in percentage of short telomeres may be more predictive of your health status or disease risk than just a baseline measurement. So the field is moving in that direction. I'm going to skip over describing this slide. It's just more data that highlights what's now known as telomeropathies or telomere disease or telomere syndrome, where individuals who are born with some of these mutations in telomerase are born with shorter telomeres even at birth, have a significantly higher rate of things like pulmonary fibrosis, bone marrow failure, liver cirrhosis, and skin disorders. Because their telomeres are short, these are subjects with disease, and this is something that we now have genetic screening tests for, but just looking at the average telomere length is often a good predictor of whether these mutations exist, and you can confirm the diagnosis with genetic testing. Okay, so short telomeres, besides doing a lot of damage, they can be used to predict a patient's disease risk in different areas. Now, telomere length by itself, because it's a global marker of what's going on in your body, it can't tell you whether it's cancer or cardiovascular disease or susceptibility to infections. That has to come from other studies. But if a person is in the shortest, or let's say the shortest 2/3 or 3/4 of telomere length, they do have this increased risk of disease. And I'll just show you some slides that describe this. So this was a big study in an Italian cohort of cancer and cardiovascular disease risk, 800 men and women, 45 to 84 years old, tracked for 10 years. And the individuals that were in the longest tertile of telomere length were the reference range, and the cumulative hazard ratios for outcomes was plotted as a function of time for this population. And if you look at cancer mortality or cancer incidence, there's significantly elevated risk for people in the mid or the shortest tertiles. That's 67% of the population. And that might reflect to this threshold length where you don't see a lot of effect until telomeres get relatively short. And then in this graph, it's looking at cardiovascular disease events and death in the study. And it's the same story, similar. In fact, the graphs look very similar to what we saw in cancer, where the longest tertile is the reference population. You see 2 or 3 or more fold increased hazard ratios in the individuals in the shortest 2 tertiles of telomere length. And then this was a recent study just published this year on COPD, chronic obstructive pulmonary disease, and mortality, looking at mortality due to cancer, cardiovascular disease, or respiratory mortality. And again, in this population, it was divided into quartiles, and those in the first, second, or third quartiles all had relatively the same risk of disease or death. Yes, this is all death. There were some disease measures, but and compared to the fourth quartile, that's where you see the ones with the longest telomeres as the reference population. So a large number of subjects actually in all of these different populations are at an increased risk. 75% in this of death from multiple different diseases. Okay, and another very interesting aspect of this, and an area where our company, Telome Health, plans to go in the future, is companion diagnostics, because almost all drugs have side effects, and they could be mild or severe, and many drugs, in fact, are now known to have an impact on either modulating telomerase activity or causing a change in the rate of telomere loss. So, for example, in cancer patients, a lot of the cytotoxic drugs accelerate telomere loss. Many of them inhibit telomerase. And so you would predict that, for example, in cancer, individuals who have short normal telomeres in their body cells, forget about what's going on in the cancer. If they're just normal telomeres as measured in circulating white blood cells or PBMCs, they may be at greater risk of having adverse effects of a telomerase inhibitor than individuals who have long telomeres. And it's going back to what I said earlier, patients often are treated with many rounds of chemotherapy, cancer patients are, or other radiation will accelerate telomere loss. So these individuals have shorter telomeres, unfortunately, due to their disease treatment. And if those telomeres are short, with a telomerase inhibitor, they may be thrown into a point of cell senescence or cell crisis in their bone marrow or other tissues and have adverse effects. So interestingly, in this study, it was statins that were impacting telomere biology, and individuals with short telomeres responded much better to statins, which have a protective effect, appear to have a protective effect on telomeres. And individuals who have longer telomeres didn't benefit, potentially because there was no need for, or no added benefit to lengthening already relatively long telomeres. So this is an area where in the future we may have thresholds where individuals, depending upon their telomere length in normal tissues, may be given different drugs or different doses of drugs in order to optimize the benefit of the therapy. Okay, I'm saying here that telomere length may be impacted by positive lifestyle changes. The omega-3 story does, from the randomized controlled studies now done, look to be something that truly can be causal in lengthening telomeres. Many other things are under investigation, and there will be controlled studies in those areas as well. Some of the animal studies show that certain compounds can activate telomerase and lengthen telomeres, and of course TA65 is one of those. So I'm just going to mention a little bit about the discovery of TA65, again supported by Noel Patton way back in the early 2000s. And what we did was, it was a telomerase activation screen against normal human cells. We looked at natural product extracts from traditional Chinese medicine. We selected herbal extracts, root extracts actually, based upon the Chinese medicine interpretation of the effects. So we didn't go after TCMs that were thought to fight cancer or be antibiotic in nature, but ones that were reputed as health maintenance or longevity extracts. And we then identified active molecules from the screen, selected the least toxic and most active compounds for further testing, characterizing them in multiple human cells and animal models. And TA65 was one of the ones that had significant activity, in fact, the best activity out of that screen. And that was what was licensed to Knoll, along with some other compounds that had similar or almost as good activity. So what happened on the drug side, because Jaron was planning to take these compounds, at least one or two of them, forward into clinical studies, was that it got very expensive. Jaron had multiple other programs in the stem cell area and in cancer, and decided to back burner the telomerase activation program as a drug. It may be resurrected down the road, but for now it's on hold. So we're very happy that Knoll is taking TA65 forward, at least as a dietary supplement. Okay, so in terms of the observational data, some of this is already published. We can't claim that TA65 is the actual cause of some of these changes that are reported in the follow-up analyses post-baseline, because there was dietary supplements given as well. So we attribute this to what was called, at the time, Patent Protocol 1. So TA65, starting out initially at roughly 5 milligrams a day, and then individuals and with their doctors' guidance sometimes escalated the dose up to as much as 100 or more milligrams per day. So the key observations post-baseline at 3, 6, 9, 12 months on the patent protocol are listed here. First of all, there was a study done by Bill Andrews and his group looking at whether the molecule in fact activated telomerase in his hands. We had shown activation in human keratinocytes and other cells at Geron, and he looked at human fibroblasts, fetal fibroblasts, which have very low but slightly activatable telomerase, and confirmed telomerase activation properties by TA65. And then in humans, we had 260 person years of use at the time that paper was published. The mean age was 62 plus or minus 12 roughly, and I think as mentioned earlier, immune remodeling was seen towards younger phenotype post patent protocol 1, a reduction in the percent senescent cells. Clearly could be no placebo effect there, or very little. The greatest effects were seen in CMV positive individuals. As expected, the chronic stress of CMV infection can accelerate telomere loss, and itself increases the percentage of senescent cells circulating in the body. And the fact that it was mainly seen in CMV positive individuals suggests that this was probably not a placebo effect, because most individuals didn't know if they were CMV infected or not. Reduction in percent short telomeres was confirmed in animals with TA65 alone, so no supplements added. That was work done by Maria Blasco's group. And then we looked at whether there was whether there was higher or lower frequency of cancer during this 260 person years, either cancer or cardiovascular deaths, and it in fact turned out to be statistically significant lower frequency of deaths, assuming that the TA65 population was same as the general population, and of course that may not be the case, but at least there was no evidence from looking at events like death or morbidity that TA65 was increasing adverse outcomes. We also saw other positive changes like increases in bone mineral density and improvement in multiple cardiovascular or metabolic syndrome markers. And that'll be subject of another paper coming out hopefully in the near future. So in terms of the animal data, some of this has already been discussed. Again, a lot of this is coming out of Maria Blasco's laboratory in Spain, really terrific model systems that are being looked at. So the bottom line is that TA65 alone increased health span of adult, meaning 1-year-old or old, 2-year-old mice without increasing cancer incidence. And I won't go into all of these details, but it was remarkably similar to what we saw in humans. So the fact that this was done with TA65 alone in mice is very suggestive that what we were seeing in the observational studies with humans was in fact truly happening and not just a placebo effect. Okay, and then in terms of the future, Noel is taking what is actually fairly uncommon for dietary supplement companies, especially small ones, to actually support randomized placebo-controlled studies. And Noel and his team, I think if Sarah Murphy is here, she's the one that's leading the effort to get these off the ground. There is a study that's going on already in a baseline group enrolled. This is the effect of TA65 on telomere length and immune system markers in CMV-positive adults, 140 individuals. So it's sufficiently powered, and it's modeled after the observations that we had from the earlier study. So can we prove it conclusively with randomized placebo-controlled studies? So there's a placebo group, low and high dose. We're going to try to get some dose response out of this. 12 months. The status is that the baseline enrollment is completed. So we're looking forward to seeing those data, and since it's blinded, we'll report the results either way. So the effects of TA65 on type 2 diabetes markers in adults is another one that's being teed up. It's in negotiation at this point, and then there's at least 10 research studies with various academic groups in multiple disease conditions. So I think the importance of telomere length testing, we've heard a lot about. I just want to have this one slide on the cancer question. So to date, none of the studies conducted by TA Sciences or supported by TA Sciences with TA65 in humans or in animals suggests genotoxicity or elevation of cancer risks. And we will, I don't have time right now, but maybe in the question period, if there's any question about what those additional studies are, I can certainly talk about it. Now, unfortunately, it's not possible yet to analyze long-term studies in humans, but to date, in almost 6,000 person years of TA65 use, at least from information we have to date, and it's not complete yet, but there's no evidence that we're seeing increased cancer incidence yet. So although TA65 could extend the lifespan of pre-malignant cells that are on the edge of senescence, we know that very short telomeres actually promote tumor initiation. As Dave mentioned, the immune system aging itself decreases some anti-tumor functions, and senescent cells in studies in conjunction with pre-malignant or malignant cancer cells, those senescent cells themselves promote tumor growth and metastases. So one could argue equally well, I would say, that TA65 could in fact, by lengthening telomeres and preventing some of these changes that happen with critically short telomeres, it could be a tumor suppressive molecule. Okay, so just in summary, telomerase, key points, it synthesizes telomere DNA, is capable of immortalizing human cells without inducing cancerous transformation, and it's regulated by genes and lifestyle. Telomere shortening is a root cause of aging and age-related disease. Activation of telomerase can reverse telomere shortening, restore regenerative capacity of cells, rejuvenate tissues. We know that in mouse for sure, and in humans, at this point, till the randomized studies come out, I would say maybe, or I might be encouraged to say likely. And TA-based interventions for healthcare actually have, in my opinion, really great potential and may dramatically improve health span.
you.